First of all, I would like to thank the organizers for inviting me to this interesting workshop and to share our research interest. So basically what I'm going to talk about is what we have been doing for the last 5-10 years regarding peptide-based vaccine strategy. We are more interested to herpes vaccine because of the morbidity and mortality that can afflict to our patients. So basically some introduction regarding herpes. So we have, as you know, two herpes viruses that share 60 to 80% of sequence, so named HSV1 and HSV2. HSV1 is mainly associated with labial and facial infections and ocular infection. HSV2 associated with genital herpes. 90% of the population worldwide is infected with HSV. 33% of adults in the US are suffering from genital herpes, and 80% of adults are shedding reactivated virus in their teeth many times a year. So in US, about 500,000 are suffering from ocular recurrent herpes diseases. A little bit about the cycle of the virus. You have a primary infection that occurs in epithelial cells. Then the virus replicate, and nearby sensory nerves it affect the neurons, and then migrate by retrograde transport to the trigeminal ganglia when it's in the head, then or to the dorsal ganglia when it's a genital herpes. Then the virus go back to the primary site of infection and induce herpetic diseases. So basically, the virus is controlled mainly by CD8 T cells through a mechanism of cytokines and granzyme perforin mechanisms. However, suddenly and spontaneously, for not really known reason, the virus get reactivated and then go back to the site of infection. We have been showing that through the latency, the virus express the LAT associated transcript that mainly have two major effects. First, that induce what we call exhaustion of the immune system. The first one is induce high expression of HLA class 1, and also higher expression of PVL1. So, and this is highly associated with the expression of LAT gene, as is shown here in this experiment. So we believe that the exhaustion is why some people have more recurrent reactivation of the virus. So I will pass this. So for the epidemiology, the majority, fortunately, the majority of the people are asymptomatic, but they are carrier of the virus. 5% of the population are symptomatic. And these symptomatic people have chronic reactivation of the virus and can have different herpetic disease, from simple lesions to fatal, and that in case of cerebral infection. So in spite of all the interventions and educational programs like sexual behavior education, barrier methods, antiviral drugs therapy, still we have an increase of HSV infection around the world. So development of an effective immunoprophylactic or therapeutic vaccine will be something important, and will reduce probably the mortality and morbidity. So as the majority of the population is infected, it's more important to develop a therapeutic vaccine for herpes. So this is just the definition about therapeutic versus prophylactic. So many formula have been made in developing vaccines against herpes, like replicated live attenuated virus, inactivated virus, attenuated viral vectors, DNA vaccines, transduced or epitope pulse dendritic cells, soluble proteins, And now what we are interested in, and I think we are pioneer in this field, is a peptide and lipopeptide-based vaccines. So these are the list. Unfortunately, all these vaccines have been stopped or discontinued, and they did bad response. So we believe that our self-adjuvanted peptide is the future to develop a herpes vaccine. So this is the strategy, basic strategy behind how to develop peptide-based vaccine. Basically, we have a protein or gene. So we select candidates epitope, and we select the highly expressed HLA in the population. Then we do some mathematical framework, or what we call algorithm prediction, and some immunological testing until we get the epitope of interest. The importance of peptide-based vaccine is multiple. This is non-infectious materials that does not have any deleterious sequence or sequence that risk to integrate in the genome or combinations. Also, we can bypass any external adjuvants with toxicity. We can design a simple or multiple epitope from the same or different pathogens, and also is soluble, safe, and cost effective. So basically, one of the advantages of peptide-based vaccine is to avoid all pathogenic peptides. And the issue here, I'm showing you how we are really doing. This is just an example how we are doing in selecting our peptides and developing at the end this peptide-based vaccine. Basically, we are doing a lot of computer predicting algorithm system for CD4, CD8. We do also HLA binding for T cells epitopes in vitro, antigenic T cell stimulation, finding out which one is recognized by symptomatic people and which ones are recognized by asymptomatic people. And also do the protective efficacy in animal model, whether it is normal wild-type mice or humanized mice expressing the HLA of interest. Then we will construct the vaccine lipopeptide. Basically, we are just using a palmitic acid. As known, palmitic acid is a very small carbon molecule that we can find in olive oil. So this is one example of peptide backbone. We put CD8 and CD4 pairs, and we try the protective efficacy in an animal model as preclinical study. So basically, in 2008, we have been the first to identify the first HLA-A2 restricted CD8 T cells epitope from HSV glycoprotein D. So these are the three epitopes. I'm not going in details of the experiments, but I'm just giving you the conclusion from the study that elicits strong CTL activity in HSV infected human and humanized HLA-A2 transgenic mice in vitro and ex vivo. Naturally processed product, because you want the epitope to be naturally also. You don't want to design a peptide that cannot be similar to the endogenously produced in real life. It's recognized by high frequency of specific CD8 T cells in HSV infected human and humanized mice. And also, it is very interesting to have the sequence homology that is shared between HSV1 and HSV2. So it will be good for HSV1 infection and HSV2 infection. Then after that, we characterize the asymptomatic human now as CD4 epitopes that are recognized by HSV1 glycoprotein B this time. So we identify 4 HLA-DR restricted CD4 positive T cells, and that elicits strong CD4 T cell activation and proliferation in HSV1 seropositive donors. 3 of them elicit strong CD4 cytolytic. It's very interesting. In contrast to mice, in humans, CD4 can be cytotoxic, like CD8 T cells. They are also naturally processed epitopes, and they are recognized by asymptomatic patient. The three dominant one are recognized by asymptomatic people, and the only one that is recognized by symptomatic. And here we got the idea that some epitopes are recognized by symptomatic people and some epitopes are recognized by asymptomatic people. And let us go for the idea of why some people develop herpetic disease and others they don't. And we find out that probably because the symptomatic patient recognize different sets of epitopes than symptomatic patients. Then we after doing a lot of animal study in HLA transgenic mice, there were this, because the best model for ocular herpes infection is a rabbit. And we developed a novel HLA transgenic rabbit model for preclinical study of our human CD8 T-cell epitope vaccine. So this is the rabbit. Why rabbit? Because rabbit have spontaneous reactivation similar to human, produce recurrent eye disease to the clinical, similar to the clinical disease, and mount human HLA, because we have selecting CD8 T cells from humans, so they recognize the same epitopes as human. So these are the characteristic or phenotypic characteristic of these rabbits. So they express HLA in the HLA class 1 in the cornea and the trigeminal conglia, and this is the non-transgenic rabbit control, and also on peripheral blood mononuclear cells. So also here, using tetramer HLA class 2, these peptides recognize the same profile in HLA transgenic rabbit as in human. And we can see that these are 3 dominant epitopes that are recognized by asymptomatic patients, are also recognized in our HLA transgenic rabbit. So then we made these 3 lipopeptide vaccine having the GD4982, which is a CD4, promiscuous CD4 T cell help that stimulate rabbit HLA rabbits, and also the 3 different dominant CD8 T cells epitope. And then we immunize the rabbit. We give 3 immunization without any adjuvant, without anything, just in cerebrolabs and PBS, and 3 immunization. Then we challenge with the virus, and we analyze all the immune function and response in TG, spleen, conjunctiva, and cornea. And also we did protective efficacy by measuring the tears collection and the viral load and disease slit lamps. So basically here we show clearly that the CD4 T cells induce T cell response in rabbits immunized with lipopeptide compared to mock immunized infected or non-immunized uninfected rabbits. Also, it induced proliferation and a high number of CD8 T cells specific in draining lymph node, conjunctiva, TGs of immunized and infected rabbits. The same here, this is the division of CD8 T cells, and the same thing, we have higher T cell proliferation in immunized infected rabbits in the draining lymph node, conjunctiva, and TGs. Also, the viral load, we can see that immunized, lipopeptide immunized rabbit have a very low titer compared to wild type rabbits, demonstrating that they have lower shading capacity compared to the controls. The most interesting slides we have is the comparison between the eye disease here, and we can see that we have a small lesion compared to bigger lesion in the eye of mock immunized, and this is completely associated with the HSV1 in the tears. We can see here that more HSV1 in the tears, more the corneal lesion are big. And here we can see the eye lesion size comparing the lipopeptide immunized versus the mock immunized rabbits. So also this is just more details with the cloudiness geographics, that we have high cloudiness geographic and iritis in non-immunized and infected rabbits compared to control or immunized infected rabbit. We found out that there is a clear correlation of epitope-specific CD8 T cell response with the protection in the eye, and it is the important things coming from here that high disease is associated with low number of CD8 specific T cells, whether regarding to these 3 epitopes in the TGs or cornea or draining lymph node. Most interestingly is the CD8 interferon gamma is mostly expressed by the conjunctiva, and we have no difference between in the cornea. So basically, when we give the ocular, the immunization, it makes a lot of interferon gamma in the conjunctiva that fight the virus replication in the eye. So this is our conclusion. The first report showing that symptomatic human CD8 T cells epitope can protect against primary HSV1 infection and disease in humanized HLA transgenic rabbit. And now we have a very strong data showing the same thing in therapeutic ocular herpetic infection. And it's also protective with a long memory response. This HLA transgenic rabbit also now it's allowing us to do that. Therapeutic immunization for latently infected rabbit with human CD8 T cells epitope will decrease HSV1 spontaneously reactivation and reduce or eliminate HSK. This unique animal model will allow us to study the efficacy of therapeutic vaccination against HSV1 spontaneous reactivation and recurrent eye disease using human CD8 T cell epitope. So the lipopeptide-based vaccine will be a very important tool, non-toxic, without external adjuvant, and to be used with selected epitopes in single multivalent vaccine molecules. So the same thing we have done regarding genital herpes, but for that we use mice and HSV2 infection challenge. And this is our lipopeptide. Basically, we have Padre, which is promiscuous epitope, CD4 epitope, working for human and many animals. And GB, and the mice dominant GB peptide. And this is the control peptide. And basically, it induces the same thing as for HSV1, strong CD8 T cell response, as shown here, with the lipopeptide. And also, it's mediated by Th1, as shown by cytokine profile production, interferon gamma IL-2 and IL-12. Also, it induces strong high number of CD8 specific T cells, and we have strong cytolytic activities. When we look at the survival and pathology mean and the titer of HSV, of course, we find out lipopeptides have the lowest pathological disease and titers, but longer survival, and so on and so on. I'm not going on details. We believe that mucosal surfaces get when we put the vaccine HTL, CTL lipopeptide, it will be catch out by dendritic cells of plasma or plasmacytoid dendritic cells. The toll-like receptor, the toll-like receptor 2 agonist, which is a palmitic acid, will get stimulated and via MYD8 transmit the signal and induce cytokine production by dendritic cells. Also, lipopeptide can be phagocytosed and processed and presented by the peptides will be presented by MHC to naive CD4 and CD8 T cells. This will generate effector CD4 and CD8 T cells, goes back and fight the HSV1 or HSV2 infection. So intravaginal delivery of the lipopeptide induce strong and long-lasting HSV2 specific memory CD8 T cells. Lipopeptide immunization is toll-like receptor and MYD8 dependent because of the antagonist for toll-like receptor 2, which is a palmitic acid. And intravaginal immunization with self-adjuvanting lipopeptide appear to be a novel mucosal vaccine approach which has attractive practical and immunological features. Thank you very much.